Well, thanks, Sarah. As my slides are coming up, I'd like to welcome everybody to our webinar today in celebration of World Down Syndrome Day. I want to emphasize a couple of things just with this initial slide. We're focused on developing therapies and understanding more about cognition and Down syndrome for both children and adults, so essentially everyone living with Down syndrome today. And the other point, as we join together with other organizations, all of you who have joined together with us today to celebrate World Down Syndrome Day, I want to emphasize the fact that it's not just about awareness, it's about action. Awareness and action together can actually make things happen. As I want to throw up the mission of Dsrtf yet again, just to emphasize a couple of points. One is the illustration of the fact that we want to accelerate the development of new therapies. This requires both basic research as well as translational research, but in addition, that this is really all about, again, just to emphasize, this is all about creating new opportunities for all individuals with Down syndrome. So what I wanted to start off with is just to give you a bit of an overview, essentially the bottom line take-home message today, that we've developed I think what we feel is a novel strategy to accelerate this process, and that we feel like we've had rapid success and validation for this new research strategy. As Sarah had indicated, more than $9 million in new research funding since 2004. That doesn't sound like a lot on one level, but as you saw the funding levels for Down syndrome research, and particularly cognition research, this is actually a significant number, and we think we've efficiently used these dollars to push this agenda and this research strategy forward. We've used this strategy to also establish critical biomedical expertise. As Sarah mentioned, we've developed a very proactive scientific advisory board, but we also encouraged the researchers that we funded to establish connections and collaborations. One thing we'd like to emphasize, too, and we think it's important for our strategy, is certainly no one institution, no one company, no one set of researchers is necessarily going to get us from point A to point B, that is the development of therapies. It's going to require interactions and collaborations and the engagement of a number of different researchers and organizations to make this happen. Again, we've defined multiple mechanisms involved in cognitive impairment associated with Down syndrome, and this has led to the identification and pursuit of at least 8 new potential therapeutic drug targets. The grants program that we've established, again, is a little bit unusual, I think, for organizations like ours, in that it targets grants. We actually proactively identified, together with the SAB, the most critical research out there that needs to be accomplished to push towards the development of new therapies. We focused on accelerating toward the innovative, safe, and effective clinical trials, together with the development of effective new therapies and new opportunities, again, stressing for all individuals with Down syndrome, not just children, but also adults. I think another significant part of our strategy has been and something that I think is novel for Down syndrome is engagement with the biopharmaceutical industry. And you'll hear more about that today, but I've just listed the 3 sets of clinical trials and engagement with different companies that have led to these landmark new clinical trials. Roche, which you'll hear much more about, Dr. Quadra will in fact go through that in more detail. Balanced Therapeutics, a clinical trial was initiated in 2012, and just recently, within the last few weeks, AC Immune, a small Swiss company that's developed a molecule that will address Alzheimer's pathology. We've also, as Sarah indicated, one of the objectives of our grants program is to develop research to the point where it's much more competitive, particularly for NIH funding and other sources. We want to encourage others to fund this research. And we've actually leveraged so far more than $7 million in additional research funding from both the NIH, universities, and other foundations. And I can't stress this enough, but this has actually occurred. We've had this rapid success, we believe, because we've established key partnerships and collaborations in achieving the breakthroughs. Again, I want to come back to why cognition research, why focus on that? Well, As Sarah had indicated, this early developmental and sustained cognitive disability are most significant across the lifespan of all individuals with Down syndrome. We think that an impact here and a focus of resources, the limited resources that exist so far, we can actually push this forward in a very significant way. And the other reason for focusing on cognition research is that it has a significant impact on the lives of individuals with Down syndrome. So there's significant related issues such as independence, speech and communication, and sleep problems, all of which relate to cognition and Down syndrome. As Sarah also mentioned, the majority of individuals with Down syndrome show the same neuropathology of Alzheimer's disease by the age of 40. And subsequently, the majority of those show earlier cognitive decline with age. Fundamental to our mission and to our strategy is that new biomedical research can significantly advance better understanding of cognition and Down syndrome, which will not only lead to safe and effective new therapies, as we're going to discuss today, but also to new and more effective interventional strategies in education, employment, and independence. So that's a primary reason of why we focus on cognition. Well, I've had a lot of people ask, well, what is cognition? It's a catch-all term, but it's actually significant. And so what I want to do is introduce you to just some aspects of cognition, which involve learning, memory, and speech in Down syndrome, and to just give you some biomedical background on the systems that are involved. So memory systems in individuals, in all of us, involve certain systems in the brain. One of those is the hippocampus, and the surrounding cortex is shown here in this slide. What does the hippocampus do? It detects and stores novel information, so actually about learning, so allowing for quicker adaptation. The more we learn, the easier it is for us to adapt to new situations. It binds together pieces of information, so the knowledge, what we've learned can be bound together and manipulated in ways to adapt to new situations. It talks to the rest of the brain to store and update knowledge. So this is a critical part of our brains in terms of learning and memory and cognition. It helps construct a map of the world in our brain. So that allows us to, for example, remember where we parked a car when we park in a parking lot. But the memories are best recalled when this map is intact. And hippocampus is one of those regions that's been shown to be involved in cognition in individuals with Down syndrome, and to show that there are, in fact, some dysfunction in that region. Another region which we focus on is the frontal lobe of the brain. Sometimes you'll hear this referred to as a prefrontal cortex. The frontal cortex is actually the brain's CEO, essentially being involved in making executive decisions. It's involved in working memory. That means it means keeping information online and being able to manipulate it and work with it. It allows for flexibility, less getting stuck on a way of solving a problem. That is to bring new strategies into solving particular problems. As I said, it helps plan the actions of individuals, so it actually is the executive function or the CEO of the brain. It regulates attention and keeps behavior in check. And finally, it's involved in abstract thinking, for example, the concept of time or things like mathematics and that sort of thing. Again, this is another region of the brain which is impacted in Down syndrome. So what does that really mean? What would we like to accomplish if we develop new therapies? It's not just some abstract notion. In this slide, what is the impact that changing these memory systems have in Down syndrome? What is this going to mean in the day-to-day lives of individuals? First, lead to more associations, greater connections in their knowledge, better attention and task juggling, better progress in school and in employment, faster processing and initiating activities. And finally, more flexibility, behavior improvement, less stubborn. This is something my family tells me that I've got an issue with as well. But nevertheless, this greater flexibility will lead to a greater ability and willingness to try new strategies. All of these are going to be things that impact the day-to-day lives of individuals with Down syndrome. Well, evidence for links between memory and learning in Down syndrome the hippocampal tasks are involved in binding information, and this relates to adaptive behavior scores. This is actually a way of measuring levels of cognition. And adaptive behavior is that behavior which allows us to adapt to new situations or to any given particular situation. It's involved in more memory for complex objects, and that relates to language ability. So this is where speech comes in. So the memory systems are clearly involved in speech abilities and capabilities. And auditory working memory is highly related to IQ, although that's not the world's greatest measure for cognition. Well, the bottom line is if we don't try, if we don't, just starting back to our mission, if we don't try, we don't know what could be. There is no language or everyday task section of our brain that can be targeted. These skills are supported by the memory systems. These are the same memory systems we are modifying in mouse models as some of the work I'm going to discuss today, such as the hippocampus and the frontal cortex, and also impacted, as Dr. Khwaja will talk about in more detail, with respect to their clinical trials and the drugs that they're investigating. Changes in these systems can have a big impact in the human, as I've said, and only through regulated, evidence-based clinical trials will we know if these drugs work and how big the impact might be. So we're very much on the cusp of answering these additional questions. Well, as part of our strategy, one of the key drivers in our strategy, and I think this is a novel aspect, is to actually take 2 steps back and look what's needed, what are the priorities, and how do we get from point A to point B. Well, successful development and approval of effective new therapies, including the essential enlistment of biopharma companies to develop and market these drugs, and their expertise requires the answer to at least 3 key questions. And these are core to our strategy. Are there evidence-based dysfunctional cognitive mechanisms together with associated validated specific drug targets and drug candidates to ameliorate that dysfunction? That's key. That's the first step. The second is, and one of the questions we dealt with very early on, which there wasn't an answer to back in 2007 when we held our first scientific advisory board meeting, is are there specific assessment tools that can measure meaningful improvement, efficacy? That is does a drug work resulting from treatment with new potential drugs, and can success be demonstrated? If we don't have such tests, there's no way to actually move new therapies forward. And finally, assuming we've answered those questions, are there potential clinical trial capable sites and patient participants that can sufficiently and efficiently be recruited for successful drug development and FDA approval? Without clinical trials, there's no way to establish a drug's efficacy or whether it's actually working or not in these individuals. As Sarah pointed out, as recently as 2004, there were no answers to any of these questions. And actually, I might add that this actually goes even several years ahead to say that we didn't have answers to these questions. Well, I won't belabor this, but just to say what we also looked at is a drug R&D pipeline. How do we go from discovery research all the way down to an FDA-approved drug? And this just illustrates a timeline. This is pretty typical, whether it's for Down syndrome or other disorders, that's required to go from discovery research through that approval process. And what it illustrates here is the expense of this actually goes up. It's not even linear. It actually goes up quite dramatically in the later stages of this. The time I've just mentioned. But one thing that's also, I think, is you read about periodically in the newspaper is that the progress of projects that are started back in discovery, as they move through this process, those projects drop out for a wide variety of reasons. Sometimes it's side effects, sometimes it's other issues with potential drugs or the mechanism. So the goal of our strategy is to decrease the expenses, decrease the time, but increase the number of projects coming in at the beginning of this process. We need to keep new projects coming in at the discovery end to make sure that we end up with successful new therapies at the end. And finally, I'll come back to this in a little bit, but just to say, when we started in 2004, the work that Sarah referred to earlier, where we were supporting work with Bill Mobley at Stanford University, all of that was focused at the very beginning here, at the discovery level. And this really projects all of this forward, summarizing what I'm going to talk a little bit more about today, to say that these various grants, you can see where they plug in this process. So what we're trying to do is to address gaps in our knowledge with high priority, as well as to identify those things that might be roadblocks going through this process and addressing those as early and as soon as we can. Well, how about answers to those questions? In question 1, we've identified at least 8 new potential therapeutic targets. These have been shown to overcome specific impairments and improve cognition in mouse models for Down syndrome. These are mouse models that have the same, quite a number of the same genes that are on chromosome 21 in 3 copies in this mouse model. And this is a major step towards the development of effective new therapies, a first step. What are those targets? Well, some of those deal with targeting the early developmental and sustained cognitive disability in Down syndrome. And one of those are looking at a mechanism impaired cognitions and communications in the brain, and we've identified 3 targets. You're going to get a copy of this. I'm not going to spend a lot of time, particularly on the GABA receptor. Omar, Dr. Khwaja is going to go into this in more detail. I'm going to skip to essentially the bottom line here and say that what we've discovered is there's a greater inhibition in hippocampus, a balance in excitatory and inhibitory neurotransmission in the brain that's necessary for learning and memory. And in Down syndrome, it's been discovered that there's greater inhibition in the hippocampus. That is, that balance there is an imbalance there towards inhibition. And what's been discovered is that there are particular proteins on these in the brain that modulate that excitatory inhibitory imbalance. And you can see that these targets that we've identified listed here, the GABA receptor, GABA B receptor, and GERT2, can all lead to improved synaptic plasticity, which is essential for learning and memory. Finally, we're targeting the earlier development of Alzheimer's disease. So we've got 4 targets that we've identified with respect to this. APP, a gene that's on chromosome 21 that's been involved in the formation of the plaques and tangles that one finds in Alzheimer's disease. That gene is in 3 copies in individuals with Down syndrome. We've actually shown that impacting that can reduce the neuropathology associated with Alzheimer's disease and also improve learning and memory in the mouse models with Down syndrome. This is actually the target of AC Immune's new drug and planned clinical trial. Finally, we've also identified another target. This is the inhibition of, or this is the restoration of another neurotransmitter that's involved in the neurodegeneration that's associated with Alzheimer's disease. And if we actually can replace that, we are able to, as shown in this slide, to either affect the decrease in APP or its products, which leads to improved synaptic function, halt neurodegeneration, improve and maintain cognitive function. But also, as I said, this modulating or increasing norepinephrine will also lead to improved cognitive function in Down syndrome. Finally, neurogenesis, the impairment of formation of brain neuronal circuits. There's a target we've identified here, the work with Roger Reeves at Johns Hopkins. This has actually shown a completely novel way of improving a specific form of hippocampal-mediated learning and memory and actually restore the development of the cerebellum. So that's illustrated here in this cartoon, which corrects the development of neuronal circuits and leads to improved motor learning and memory formation. Questions 2 and 3. These are translational initiatives that address those gaps and specific roadblocks. Down syndrome specific cognitive test battery. The Arizona Cognitive Test Battery, which we supported with Drs. Jamie Edgen and Lynn Adel at the University of Arizona, they developed actually the first Down syndrome specific cognitive test battery. And this can significantly enable the determination of whether the drugs work in clinical trials, as well as be useful in understanding more depth about cognition in individuals with Down syndrome. Also, what about clinical trials and participants? Well, we've been funding the Down Syndrome Cognition Project at Johns Hopkins with Roger Reeves and 9 different institutions, probably 13 additional principal investigators around the country. What this is actually establishing and creating a scaffold for effective Down Syndrome Clinical Trials Network. So we've been able to address these questions, those 3 questions sufficiently, and that's been particularly useful. Those were necessary elements in engaging the biopharmaceutical industry. Again, I'm not going to spend a lot of time on this, but to say that this led to a landmark initiation of a major new clinical trial in September of 2011. Dr. Khwaja is going to speak about that in somewhat more detail, so I'm not going to go through that further at this point, but say just to say that they now have 3 clinical trials ongoing in Down syndrome, which he'll describe in more detail. We've also engaged with Balance Therapeutics. This is a small biopharmaceutical company, and they initiated a clinical trial in August of 2007. This is a drug, this is actually PTZ, which you've heard about in some of our previous webinars. It's an investigational drug. It's targeting, again, the amelioration of that inhibitory-excitatory imbalance and addresses overcoming the cognitive behavioral impairments in individuals. This is in a phase 1 trial currently in Australia. Finally, as I'd mentioned, the most recent addition to this is AC Immune. They received FDA approval for an IND, investigational new drug application for a new clinical trial in adults with Down syndrome. This is actually an investigational drug targeting the amelioration of Alzheimer's disease neuropathology in individuals. So it's going to address the reduction of Alzheimer's disease plaques and overcoming the associated memory impairments in individuals with Down syndrome, hopefully halt that process. Finally, just to summarize, again, we've got limited time today, but just to say that these are the current grants that we're doing. These are continuing to push forward those that research that I had just mentioned earlier. And what I want to do, without being able to go through this in detail, is to say I've included here a URL to our website, which will describe in more detail the ongoing research that our grants are funding. This is, again, pushing the envelope forward on all of these fronts, and you can see that this is addressing multiple points throughout the drug discovery research and development pipeline. Well, not only have we supported grants, but we actually take a proactive and strategic role with NIH and Congress. The idea there is to enhance new Down syndrome research, maximize the synergy, and increase federal funding. While I don't have time to go into grand detail, we are participants in an NIH Down syndrome consortium with other Down syndrome organizations with the NIH. This is to be able to create dialogue and with the NIH to, again, affect new research, develop synergy. Part of the activities there, it's a Down syndrome patient registry. The launch goal for that is mid-2013. There are actively two boards working on that together with the NIH funding and researchers on developing this registry, a governance board and an operations board. And I am happy to say that I'm actually a participant on the governance board. You'll learn more about that as this gets closer to mid-2013, and hopefully we'll have a webinar focused on that. The other is an NIH Down syndrome strategic plan, which was developed back in 2007, 2008, and 2009. That's under the process of review for an update, given all the progress that's actually occurred. Well, why is this important? I mean, one take-home message and one thing that you can communicate more widely out there, why is any of this important to the wider community? I've already indicated that about Alzheimer's disease. Well, because of that shared neuropathology, some of the development that's going on in Alzheimer's disease drugs out there certainly will, the individuals with Down syndrome may benefit from that. But significantly, for the same reasons, the work on research in Down syndrome and Alzheimer's disease and the development of new drugs to treat AD can have a wider impact for the wider population. I won't belabor this, but solid tumors, there is evidence that there is a lower incidence of a variety of solid tumors in Down syndrome. Understanding that will very possibly lead to a widely applicable new therapies for solid tumor cancers for all of us, just beyond the Down syndrome population. Atherosclerosis. Again, research suggests that there's a lower incidence of atherosclerosis in Down syndrome, and this may lead to effective new therapies. So, again, another take-home message here is the individuals with Down syndrome are uniquely contributing to all of us. It's beyond just the Down syndrome population. How can you be involved and make a real difference? I mean, all this progress has been tremendous, but how do we continue this? Well, continue to become well-educated supporters and consumers of evidence-based Down syndrome biomedical research. We should be demanding the highest quality research that's available out there. And one way we think our strategy has shown that we can accomplish this and to speed this process along is partnership together. because we can leverage the limited resources that are available and accelerate the realization of effective new therapies and new opportunities for all individuals with Down syndrome. Critical need for participation in validated evidence-based clinical studies. I've mentioned the drug clinical trials. Omar will talk more about that in just a moment. But the point is, if we don't have participants in these clinical trials, we won't be able to push this forward. We won't be able to answer the question, do these drugs work and how big of an impact can they have? I've mentioned the Down syndrome patient registry. That'll come online by mid-year. Encourage people to be involved in that. It's going to be a way of getting feedback to the community about biomedical research, but also to inform and provide opportunities for individuals to participate in clinical studies and trials. I won't spend a lot of time talking on this, but there are other studies out there. There's a Down syndrome heart project, a Down syndrome cognition project, looking at variability in cognition in individuals, which we've been supporting for several years. They also need participants. Well, to summarize, we believe this is actually now a seize-the-moment opportunity, unusually significant opportunity at this point with the progress that's been made. This is tremendously rapid progress. The results and progress achieved to date signify that effective new treatments are within reach for people with Down syndrome. Other take-home message is based on all of this research I've described that we've put together with these different groups, understanding and treating Down syndrome is no longer too complex or difficult, and it's not too late. Cognitive function can be modified even in adults. There's a compelling case for significant proportionate increase in funding and investment in more fundamental and translational Down syndrome research. Look how far this has gotten us on a relatively small sum of money at this stage of the game. So there's high efficiency and greater promise, and we think this justifies significantly increasing the funding. So, but also emphasize, while I've talked about a lot of research today, there's significantly more promising and needed new research that we look at all the time than current resources available to participate. And finally, just to emphasize, again, one point. Our success so far, I think, has come built on collaborations and partnerships. And this requires building upon and increasing cooperation, collaborations, and partnerships, researchers and clinicians and their institutions, the Down syndrome community and organizations, federal agencies, and including all the NIH institutes, as well as engaging with biopharma companies. This can make this happen, and that's what it's going to take to speed this along. So we're building new momentum in Down syndrome research, which is creating new opportunities for individuals, children, and adults to further realize their dreams. Join together with us in partnership and be part of these breakthroughs. I thank you for your time today, and I'd like to take the opportunity to introduce Dr. Omar Khwaja. He's a translational medicine leader at Roche Pharmaceuticals and oversees the clinical trials in Down syndrome. As I said, these are landmark clinical trials. We've been engaged with them for some years. Answering those questions allowed me to meet with Roche, I think beginning actually in 2009, several years ago, and work towards developing these clinical trials. We continue to be engaged with Roche in leading to hopeful success for these clinical trials. So with that, I'll turn over to Dr. Khwaja.
Thanks very much, Michael, and thank you everyone for attending this webinar. I'm just going to try and make sure I can move the slides. So, as Michael said, I'm a translational medicine leader for neuroscience at Roche, and essentially what that means is I'm a pediatric neurologist by training, and I'm responsible for the clinical development of molecules that are relevant to conditions like Down syndrome within the company in terms of the early development when a molecule moves from the laboratory into human studies. So what I'd like to do is talk to you just about one of the targets that Michael alluded to, one of the 8 targets that so far have been identified as potentially treatable, as potentially usable as a drug target, and talk to you about the process of developing a new molecule for treatment of intellectual disability associated with Down syndrome. One of the things that we are doing internally at Roche is to recognize World Down Syndrome Day today. And before I talk about molecule development, what I'd like to do is just talk to you about our vision. And our vision is very closely linked with the statement of the Secretary General of the United Nations, which is that all individuals living with Down syndrome can achieve their potential and make an important contribution to society. And we see ourselves as a pharmaceutical company as a partner in that. So the Down syndrome team at Roche has a vision, and that's to develop a therapeutic option for people with Down syndrome to improve their cognitive abilities and adaptive behavior, and so enable a more independent life. So just to give you a little background on the company, first of all, so Roche is headquartered in Basel in Switzerland. It was founded by the Hoffman and the Roche families, and they still hold a majority stake. It employs about 82,000 people, and it's currently active in 150 countries on all continents. And here in the US, we have our headquarters are Genentech in South San Francisco. We're very focused on healthcare, and we're leaders in pharmaceuticals. We're considered one of the 10 big pharma companies, and it's the world's largest biotechnology company with medications in a number of indications, but including the central nervous system. And because the family still holds a majority stake, and because Roche is very focused on healthcare, we have a unique innovation model, which is that we have two organizations, one based at Genentech and one based at Roche in Basel, which lead the discovery and early development of molecules. And then once those show proof of efficacy and safety, then they move to the Roche late-stage organization that conducts the later studies and eventually registration of the drugs. So what does that all mean? So one of the things that I think it's useful for people to get an understanding of, because it gives you an idea of how important the work that the organizations like the Down Syndrome Research and Treatment Foundation do, is because drug development, and particularly in neurological disorders, is very lengthy, it's very expensive, and it's very time-consuming. And by partnering with organizations like Dsrtf, we get very significant input and advice, which allows us to attempt to accelerate these programs and increase their chance of success. So what this slide shows is the typical drug discovery and development process. So it begins with identification of compounds that may target your disease area, and these can be several thousand or hundreds of thousands even. And through a process of testing and development in laboratory studies as well as in animal studies, eventually these get narrowed down to some clinical candidates. And then these go through very extensive toxicology testing, in other words, to look at their safety in animals before they go into humans. And then they enter what's called early clinical development. So they first of all go into a small number of healthy volunteers. So these are people without the target condition, to make sure that the drug is well tolerated and is safe. And then they enter what are known as phase 2 studies. And phase 2 studies are clinical studies of the medication in people with the condition to see whether there is any evidence of benefit, as well as to see whether there is safety. Once the drug goes through this, they then go into very large phase 3 trials where the drug is given to people with the condition, but to lots of them to see if there are any other findings as well as to confirm benefit. And then once this is done, package is submitted to the country's regulatory, drug regulatory agencies. So here in the US, that's the FDA. And the FDA reviews all the data that's been accumulated and make a decision whether or not to approve the drug. But the process doesn't stop there, because then the company is involved in what are called phase 4 studies, which are surveillance of the drug in the community to see how it's actually performing in people who are taking it on an everyday basis. Now, to give you an idea of the scale of this, so first of all, the typical timeline for development of drugs is up to 12 years nowadays. And the number of molecules that are successfully taken into approval in phase 4 studies is very, very low. The cost of developing one drug, particularly for drugs that are involved in modulating the nervous system, is estimated at somewhere between $800 and $1.7 billion per drug. So I think that gives you an idea both of the scale of the investment from companies, but also about the need to partner with organizations to try and reduce the timelines and to increase the probability of success of these type of trials. So this is just a schematic, a diagram that shows you all the different steps. So preclinical studies, the company decides that they want to develop a treatment for a particular condition, and a research team is formed. New chemicals are synthesized and tested for safety and efficacy in test tubes in animals. And then that compound, if it shows safety and efficacy, then has to be worked up to make sure that it can be scaled up in sufficient amounts which are pure enough for use in humans. The company then files an application to the FDA to begin human studies, and these human studies start in healthy humans. And then phase 2 studies are done to look at the effectiveness of the drug in people with the condition, and then phase 3 studies, and then finally the company files all that data with the FDA and hopefully is approved. So why Down syndrome and why cognition in Down syndrome? Well, the reason is for Roche, as an innovation-driven company, is that we really try to go where the science is. And one of the things that's really happened in the last 5 to 10 years, in part due to efforts like those that you heard described by Michael, in terms of understanding the biology of Down syndrome, is that we've realized that people with Down syndrome have very specific strengths and they have very specific impairments in areas of cognition. And one of these are related to the functions of the hippocampus and the frontal lobes. And our goal is not to cure Down syndrome, but is to provide a treatment where the biology tells us that there are specific areas that could be improved through modulating those parts of the brain. So this is what's called rational drug design. So in contrast to older types of drug development where there was just screening of libraries and a chance finding that a drug might be effective in a particular disease, what we're really now looking at is taking a human condition and trying to identify the genetic basis and then model these in animals, understand the biology or the cellular pathophysiology, and thereby identify targets that could be potentially modulated by a drug, and then develop a molecular therapy that improves that aspect of the human condition. And so this is a very precise and rational way of developing a drug, as opposed to just screening a large number of compounds and hoping that you find some that may be effective in whatever condition. And that's really why Roche is involved in research in Down syndrome, because we believe that the biology has advanced enough for us to be able to understand the brain circuits that underlie some of the cognitive impairments that people with Down syndrome commonly have, and that those are potentially things we could improve by developing a drug treatment. And central to that has been the development of animal models, and this is just one of a number. But this, the mouse that you see on the left-hand side is what's called the Ts65bn mouse that was developed by Muriel Davidson at the Jackson Laboratory. And this mouse is a very useful model for studying the biology and potential effects of new molecules of Down syndrome, because this mouse has partially replicates trisomy of the mouse equivalent of human chromosome 21. And because of that, the mouse appears to have mouse equivalent, a mouse equivalent cognitive profile that we can translate into humans. So, for example, this mouse has very specific problems with a type of memory called episodic memory, and that are related to the hippocampus as well as to the frontal cortex that Michael talked to in his seminar. So the bottom line from this slide is that this mouse models many of aspects of the condition of Down syndrome, and that we can therefore use it as a useful tool to try and understand both the biology as well as for testing compounds that could improve deficits. So basically what this slide shows you is that taking people with Down syndrome as a starting point, what we understand is that they don't have globally impaired cognition. What they have is very specific impairments in cognitive function that's related, for example, to the hippocampus, and that we can use the Ts65bn model as a way of exploring that more in ways that we obviously couldn't do in people. And that allows us the mouse allows us to study behavior, but also the physiology, in other words, the actual functioning at a cellular level in the brain, understand how the circuits in the brain are different in the trisomic mouse, and this finally allows us to identify potential targets for drug therapy. And drugs usually work on receptors, in other words, parts of cells that drugs can bind to. And as Michael said, there are a number of different approaches to and different targets that we might expect to be beneficial for treatment of cognitive function in Down syndrome. But the one that we specifically focus on is a chemical system known as the Gaba system. Now, the way that the brain has billions of brain cells, and these connect to each other with trillions of connections to each other, and the connections are known as synapses. And there are many different types of synapses. But what a synapse is a junction between one nerve cell and another. And the way that nerve cells talk to each other is to release chemicals called neurotransmitters. And there are a number of different neurotransmitters in the brain. You may have heard of some of them, like acetylcholine or dopamine, but one of them is known as GABA. And GABA, we believe, is the major inhibitory neurotransmitter. In other words, when a nerve cell generally releases GABA onto another nerve cell, what it does is it decreases the electrical activity of the receiving nerve cell. And what we are coming to understand is that effective learning and memory requires a balance between this inhibitory activity that GABA is responsible and excitatory activity, which is mainly related to a neurotransmitter called glutamate. And so to have effective learning and memory, you need these two systems to be balanced, and particularly within the hippocampus and within the frontal lobes. Now, in Down syndrome, what we believe is happening, at least in part, based on studies which have been done in mouse models, for example, is that there is excessive inhibition by GABA neurons. In other words, there is too much inhibition by GABA, particularly within the hippocampus, and that this underlies the problems that the mouse has, and presumably by extension that people with Down syndrome have, with these specific forms of hippocampal memory and learning. So why is Roche particularly interested in the Gaba system? Well, Roche has been involved in development of drugs for a long time, and a very, very early drug that was developed at Roche is a well-known one known as Valium. And Valium is a drug that binds to the GABA receptor, and if you know about Valium or other medications that bind to the GABA receptor like barbiturates or alcohol, the effects of these type of drugs that stimulate the GABA receptor is to effectively cause a decrease in anxiety, to cause sleep, to have a calming effect on the brain, if you like. But what we're really interested in is the opposite of Valium, the anti-Valium, in other words, so a drug that would block the GABA receptor and release this type of inhibition. And because Roche has been involved for a long time in understanding studying the GABA receptors, this is where we decided to focus. So Roche's interest in Down syndrome comes from two things. One is an understanding of the biology, or a better understanding of the biology of cognitive problems in Down syndrome, and that one of them appears to be too much GABA in the hippocampus, too much effective GABA in the hippocampus and the frontal lobes, and the fact that we have a large chemistry program that has developed molecules that can target the GABA receptor, either to stimulate it, as in the case of drugs like Valium, but also newer drugs which actually, in effect, can block it. Michael talked about some of the science, and one of the things that was very important for us was this paper that came out by Fabian Fernandez and colleagues. And this basically showed that it was possible to use a type of medication that blocks the GABA receptor, and that would improve the cognitive impairment that the mouse model of Down syndrome had. And this was a molecule called PTZ that Michael mentioned also. And so what these show is the light gray bars are wild-type animals, or animals that don't have trisomy of the mouse chromosome 16, which is the equivalent of the human chromosome 21, and then the dark gray bars are mice who are trisomic. And when the mice are just fed milk, the trisomic mouse has much more difficulty with a number of cognitive tasks, but one of them here is object recognition. And what Fernandez and colleagues showed was that if you gave this GABA blocker called PTZ, that you were able to reverse that, and so that both the trisomic mouse performed as well as the mouse that didn't have trisomy. Now, PTZ is a drug which is in clinical trials in Australia and New Zealand. Roche decided that they also, because of their experience with GABA receptor, was that we had compounds that potentially could also do the same thing. And so what we have is a new drug called RG1662. And the reason that we are looking at this particularly is because this, again, targets the Gaba system. It's, in many words, in some ways you could think of this the anti-Valium. But unlike PTZ, which is non-selective, or other compounds like that, this one is highly selective. And what we believe that is that potentially can improve the profile of the drug and make it much more selective for treating cognition. And we went on when we did phase 1 studies and conducted safety and tolerability studies in people without Down syndrome, so these were young volunteers. And that basically, the results of those studies suggested to us that the drug is safe enough to go on and conduct clinical trials in both adults and teenagers with Down syndrome. And eventually what we hope to have is enough data to allow us to go down into even younger age groups, probably starting around age 5 or 6. So the RG1662 is what's known as a highly selective GABA negative alpha steric modulator, which is a very, very long name to say that this is a drug that targets just one subunit of the GABA receptor, and so we believe it's the subunit that's responsible for cognition and memory. And we just recently published the paper that showed the preclinical results and showed that using an equivalent drug to RG1662, that we could rescue the cognitive problems that the Ts65bn mouse has. And this is a very important paper, and perhaps at some point what we'll do is try and provide to Dsrtf, if we haven't done so already, a more easily understandable summary of that data. But basically what this showed is that by using this compound, by studying mice that are trisomic as well as mice without trisomy, that we can essentially make the trisomic mice have learning and memory functions which are equivalent to the mouse without trisomy. So we're currently conducting 3 clinical trials, and Dsrtf has been a very early partner with us, and I think many of the other initiatives that Michael talked about are going to be very key to making sure that we develop drugs safely and effectively for people with Down syndrome, and otherwise knowing where people are, that people have an understanding about clinical trials and whether or not they should enroll in them, and also understanding what are the best measures to measure the effect of the drug or the benefit of the drug. So we have 3 studies. They all have long names or long numbers. So we usually refer to the first one as a drug study in people with Down syndrome. Then we have another one which is a non-drug study in Down syndrome. And then we have a third study at the moment, which is a PET study, an imaging study in people with Down syndrome. So what I'll do is just go through these 3 studies briefly. So the drug study is to assess the safety and tolerability of RG1662 in young adults aged 18 to 30. And we're also trying to understand how well it's handled by the body. In other words, how is it absorbed, metabolized, and excreted. And then we're also trying to explore the effect of the drug on cognition as well as biomarkers, in other words, measures, objective measures, which tell us whether or not the drug is having an effect. So that study is ongoing. We're recruiting men and women aged 18 to 30 with Down syndrome, who are able to give assent and have verbal skills that allow them to participate in the assessments, and if they're on medications, that they are stable on those medications for 8 weeks prior to the study. And what we're looking at in terms of the cognitive measures are measures of attention, memory, language, and adaptive function. In other words, performance in terms of everyday living skills. That study, that study is ongoing at the moment in the US, and perhaps Julie could provide a link to clinicaltrials.gov if you want more information about the study or the sites where it's being conducted. The second study is a non-drug study, and this is an extremely important study that's assessing the performance and reliability of the scales and measures that we would use to assess cognitive function, memory, adaptive behavior, activities of daily living, and impact on caregivers and families in large studies of RG1662 in people with Down syndrome. The study actually is ongoing, was done in France, Spain, the UK, and the US, and recruited 60 people, and we're now in recruitment of the final 30 people in Canada, in Italy, and Argentina. And the reason that we're doing it in so many countries is we want to understand the impact of the cultural environment and language, as well as standards of care in different countries, to understand how they impact these measures. So this study is recruiting teenagers and adults with Down syndrome aged between 12 and 30. And there's no drug involved. It's just 3 visits to assess a number of these measures that we would use over 6 months. The final study is a study that's happening in the UK, and this is a PET or positron emission tomography study together with fMRI. And the goal of this study is that we did PET studies in people who don't have Down syndrome, and we understand well that the brain gets into the brain, that the drug gets into the brain, and that it binds those parts of the brain that we believe are important, so the hippocampus and the frontal lobes. What we're trying to understand through the PET study is whether that's the case also in people with Down syndrome. We also want to know whether people with the brains of people with Down syndrome have the same number of Gaba receptors in those parts of the brain where we think the drug works. And then the fMRI portion is to help us study by imaging whether giving the drug to people with Down syndrome changes, gives a signal of change in the brain. In other words, that the drug is actually having some effect as well. So this study is just recruiting in the UK, and again, we can provide the link to that if there are people who may be able to participate in that. So this study is recruiting young men and women between 18 and 30, both with Down syndrome and without Down syndrome. There are a number of reasons why you can't participate, but this is an extremely valuable study because it will give us very important information about the binding of the drug in the brain of people with Down syndrome and how similar or different that is to people without Down syndrome. One very important thing, though, is although these studies sound very detailed, what we're really focused on is what the impact of any drug may have on the overall quality of life with people with Down syndrome. Because while we might have a drug that improves cognition or language, what we don't want is a drug that would have a negative impact on any other area of functioning. And so all of these things are part of what we call the outcome measures or endpoints that we want to study. And so these include emotional and personal functioning, psychological, social functioning, the impact of therapy on how satisfied people are who have Down syndrome as well as their carers, and any impacts positive or negative on their overall health status. So where do we ultimately, if this molecule or other molecules like RG1662 are successful, where do we feel that they're going to fit in the overall care of people with Down syndrome? And ultimately, even though it's a very big investment and it's taking time and a lot of effort to develop, ultimately any drug that potentially can improve cognition or memory or language will still remain a very small part of the overall picture of what people with Down syndrome need, and will never replace things like speech therapy, education, physical therapy, occupational therapy, hearing and vision assessments and making sure that people with Down syndrome who have hearing impairments or vision impairments have appropriate supports for that. So this is a drug that we hope will enhance other types of therapy, like education, schooling, speech therapy, that will maximize the potential of these therapies to bring benefit to people with Down syndrome. So this is not a drug that will ever completely replace any of these things, but what we hope is that it will improve the chance of success of these by targeting those brain systems directly that appear to give rise to this different cognitive profile that people with Down syndrome have. So just to give you an idea, because I think when people from industry often people just visualize this big faceless company. In fact, there's a large team, and so this is some members of the team here in Switzerland. So I'm there on the right-hand side, and the project team leader is standing next to me, Dr. Jana Nildeker, and there's a whole group of very good and energetic people who want to join with you all in recognizing World Down Syndrome Day, and who are participating in the studies that we're doing. So I want to thank you and to thank Michael and Dsrtf for allowing me the chance to speak and talk about just one example of the impact of activities that Dsrtf has in promoting therapeutic development. And what I'll do then is finish now and then take any questions that people might have. And thank you.
So thank you, Dr. Khwaja. I think we've had a great overview today of all the exciting things that are going on, and we have a whole bunch of questions here, so we're going to get to that in just a second. Before we do, I just wanted to remind everybody that Dsrtf has a really exciting match going on today in celebration of World Down Syndrome Day. You can donate towards the match at our website. And with the 3 to match, just a reminder that, you know, a donation of 100 can become 400, 500 becomes 2,000, and so on. Right before we got on the webinar, I got an update that we had raised over $50,000. So with the match, that's over $200,000 today. To put that into perspective, we know that $1,000 buys 2 mice in a lab, or a day of research at all of our combined research institutions. So this is really exciting to see, and I welcome everybody on the call and everyone they know to participate in our match. With that, I'm going to pass this along to Julie Robichaux. Julie's helping us throughout the webinar today. She manages all of our marketing efforts, and she will tee up the questions that you guys have raised throughout the webinar.
Hi, everybody. I would like to pass on some questions, and I'd like to ask Dr. Khwaja and Dr. Hartbold just to jump in when you feel the question is your particular purview. The first question we have is from Laurie. She asks, is the GABA receptor associated at all with the presentation of ADHD and the mechanism of action in Ritalin-type medications? My understanding is that ADHD also involves a neurotransmitter problem. My 13-year-old son has Down syndrome and ADHD.
Yeah, that's a really great question, Laurie. And so the GABA receptor is not known to be specifically related to the presentation of ADHD, and the mechanism of action of medications like Ritalin really work on another neurotransmitter system called dopamine. But I think one of the things that you raise in your question, talking about your son who has ADHD, is that when we are targeting neurotransmitter systems in the brain, what we really have to understand is not just the direct impact on those neurotransmitter systems that we're targeting, so in our case the GABA receptor, but because the brain is an interconnected whole, is whether it would have impact on other brain functions, both ones which are pathological, like conditions like ADHD, or just normal brain functioning. And that's why clinical trials have such a large safety aspect to them, because we want to understand the impact of our drug, not just on the target, but also what are called comorbidities, so other conditions that are associated with Down syndrome. So that's a great question. So, I mean, the presentation of ADHD is still not fully understood, but at least through the effective treatments like stimulants such as Ritalin, for example, we believe that the dopamine system is actually the one that has the most impact.
I might just add one of the other systems I didn't have time to go into grand detail, but some of the work that we're continuing to fund at VA Palo Alto with Dr. Ahmad Salehi is focused on norepinephrine and restoration of norepinephrine levels. There is, in addition to dopamine, as Omar just mentioned, there is an involvement of norepinephrine in that system. So to some extent, while we're not looking directly at ADHD, there are some behaviors that are associated with cognition that are related there, and that work is actually ongoing and focused in that area.
Thank you. Our next question comes from Jason, who asks, has any study looked at whether there's a difference in taking a drug before versus after the onset of Alzheimer's symptoms? If so, was there found to be a difference?
There have been, at this stage of the game, there haven't been a lot of clinical studies looking at Alzheimer's disease with specific drugs in individuals with Down syndrome. There are some studies looking at drugs that have been used in Alzheimer's disease, some of the acetylcholinesterase inhibitors like donepezil or Aricept. There have been some small clinical studies there. The results have actually been sort of equivocal and looking at sort of before and after. So it still remains a hazy picture there. And I think the bottom line for those studies so far is those drugs don't look to have necessarily a very robust effect. They do show some improvement, and that's why they're used in individuals with Alzheimer's disease, but it's been difficult to prove or establish a positive effect in this few clinical trials that have been done in individuals with Down syndrome. So the answer, I think, to your question might be no.
Thanks. We have a question from Mikai, who wonders about the new drug by Roche, whether it'll be administered at different times of day, considering the recent findings from Dr. Heller about circadian rhythms and different effects of the drug based on time of administration.
That's an important issue. So the way that drugs are developed is that we really look very carefully at the profile of the drug once you take it, in this case by mouth, and then we look how the drug enters the blood, and then the level of the drug rises in the body and then falls. And so far, what we understand from our information that we have so far in healthy volunteers as well as in young adults with Down syndrome is that in order to maintain an effective level of the drug is that we would give it twice a day, approximately 12 hours apart. Now, the research that Dr. Heller is doing as well as other people, I think, is emerging information that we will have to take into account, which is this is a very new area. In other words, the influence of circadian rhythms on how the body metabolizes any drug, for that matter, whether it's Motrin or Tylenol or more complex medications. So it's certainly something that we keep an eye on. But the data that we have so far suggests that we need to do dosing twice a day, separated by about 12 hours.
Okay, thank you. We have another question. This one from Patricia, who asks, how soon do mouse models receive the drug? How soon do you notice neurological differences versus behavioral changes? And will medication need to be a lifelong treatment?
Okay, sorry. Go ahead, Omar.
I can only speak for the Roche molecule. So we've done different strategies, which is to treat adult mice as well as young mice, so mice before they reach adulthood. And the way that we designed our studies is that the mice were treated for approximately varying from about 6 weeks to about 5 months, and then they were studied for their behavioral changes. So the short answer is we don't know how quickly beyond that we would see neurological differences, and those studies are ongoing at the moment. The other question that you raised is very important. Will medication need to be a lifelong treatment? And that's one of the goals of our clinical trial studies, which we're doing or that we will be doing, which is to really understand whether there is a point at which the drug reaches its maximum effect and you no longer need it, or whether you can take the drug and then stop taking it and you still have benefit from it. So we don't have the answers to it, but those are things that we are that we're actively exploring both in the mouse models as well as in our clinical trials in people with Down syndrome.
You know, I might just add there, I think it's going to depend on the drug and the mechanism of action and ultimately what the endpoint is. And just to give you a couple of quick examples is to say that the work I had mentioned earlier with the SHH activator that's been being undertaken by Dr. Roger Reeves at Johns Hopkins. In that case, he has the particular experimental drug, and this is not advanced to a clinical trial as yet. In fact, I'm just going to underscore what Omar said. We just really don't have the answers to that question in humans at this stage of the game. But in the case of this particular activator that Dr. Reeves has used, that's actually been, what he's shown in the animal model is a single dose at the day of birth. First of all, after approximately a week to 2 weeks, and this is the mouse again, the cerebellum is restored to normal development. The other thing is with that one dose, he's actually looked 2 months later and sees a positive impact. That's when they actually measure the effects on learning and memory. So that one dose, day of birth, in these mouse models, look at 2 months later at impact on learning and memory, and you see the improvement. In contrast, I'll just mention the AC Immune study. I didn't have time to go into that, but that actually ends up being a vaccine against some of the toxic elements that go to form the plaques in Alzheimer's disease. So a vaccine, it's going to be, you can imagine that to be something similar to vaccinations for various infectious diseases and that sort of thing. So that the idea there would be that you'd be administered this vaccine. It's not clear whether or not this would require a boost subsequently or not. But that just shows you, I think, the variation that you might see between different drugs, different endpoints, and we still don't know the answer in humans. That's going to be part of the work for clinical trials.
Thank you for all your questions. We have several people asking how they can find out more about participating in clinical trials.
Well, one thing, I'll just jump in and say that on Dsrtf's website, we actually post links to the clinical trial site and basically how to get there. We've done that in our newsletters as well if you've been signed up for our newsletters. But there is a government site which Roche and all the companies actually participate in listing clinical trials for various disorders. It's called www.clinicaltrials.gov, G-O-V-E. If you go to that and you enter in a keyword Down syndrome, you'll be able to see the various clinical studies. And I know all three of the Roche trials that Omar mentioned are listed on that. The others the other clinical trials aren't yet listed on that, but we have, I think I've included in my presentation, which you can get copies of subsequent to the webinar, the URLs or links to those clinical trials.
So I'm going to jump in here. We've gone a little bit over our time, and I just really wanted to thank everybody on the call for listening in. I want to thank Omar and Michael. Thank you so much for sharing all this great information. I know we have a lot of questions here we didn't get to, and so I invite those of you that didn't get your questions answered to email us at dsrtf@dsrtf.org, and we will be sure to get those questions answered. Also want to just let you know that this webinar will be posted alongside all of the webinars that we do monthly on our website, so you can review it if you didn't catch all of it or share with friends and family. So thanks again to all of you, and happy World Down Syndrome Day. Thanks.
Thank you.
Can I just add, Sarah, just to say, I think, Julie, correct me if I'm wrong, but copies of the presentations will be available if requested, too, at the email address you mentioned.
That's great.
Yes, we'll make sure that copies of the slides and the presentations are available. Thank you.
Thank you.
Thanks again, all.